Great, thank you. So hopefully I'm gonna, oh, I guess I have to advance to make slides. Excellent, this is how this works. So I am a clinical geneticist at Boston Children's Hospital and faculty at Harvard and the Broad. And I really work on how can we improve rare disease diagnosis as an important step towards knowing what therapies and what treatments and what management patients are going to need. And so I'm going to try to convince you why this is an important question, because this is, I know we come from a lot of different backgrounds here. And so even with all of the advances in technologies that we have, the majority of rare disease patients actually remain undiagnosed. And some of this is because we actually don't understand what most genes do. Of our 20,000 human genes, only about 5,000 have been linked with disease. And even in those 5,000, we don't understand many of the variants that actually impact the function of the gene. And obviously there's another 15,000 genes that could be linked with disease that we just don't know yet. And one of the reasons I think this is a really interesting thing to study is that in these patients, when we disrupt a gene, we can see the impact of the gene on biology and by seeing what phenotypes they have. And so this has been done in many different ways. I'm showing you an example here of hypertrophic cardiomyopathy. We really understood a lot of the genes and their role in the heart just because they're found to be mutated or have they have variants with altered function in athletes and others with hypertrophic cardiomyopathy, things that we might have found through other studies, but really can almost use like humans and treatment of their diseases as a sort of natural history kind of way of understanding conditions and the role of genes on phenotype. So why do patients need these diagnoses? There's often not a direct therapy or cure for most rare diseases. Only about 5% have something that we can really intervene on. But still, it can guide medical management. We can understand more about the complications that might occur. We can monitor and screen for things. We can assist in family planning. It just provides psychological relief. It's very, I mean, we all, like, having multiple medical problems kind of happen and not understanding why, whether to yourself or to your child, is very stressful. And understanding that there's the underlying cause and that what goes together is actually very helpful. And a lot of parents feel guilt about their child is sick. And so like understanding that there's a genetic reason for this just gives them a little bit more empowerment. It allows them to use that to connect with others who are struggling with these same very rare diseases. And then they can participate in research if they want to learn more, even if there aren't direct therapeutic options. And again, we're hoping to have more therapeutic options over time. And so that's for an individual, a single diagnosis for a single person in our family. But actually, there's a lot of it that's important about diagnosing everyone, large populations of patients with rare disease, because then they can build these communities. And from these communities, we can start to understand what variants in genes are pathogenic and what are benign, and we can really start to better understand gene function. We can understand disease biology. We can develop these therapies as these communities grow. And one of the other things we've been trying to work with rare disease organizations to do is to estimate genetic prevalence. And so in work led by Samantha Baxter here at the Broad, we have developed GENIE, or the Genetic Prevalence Estimator. And I'm showing you here, let's see if this does work. Not so much. Okay, I'm showing you here using the NOMAD database, so people in the general population, as looking at carriers for recessive conditions, and then taking what we know about the disease variants and looking across different ancestry groups and trying to estimate how common these rare diseases are. Because these are generally very rare, and they're not, the medical record system, the medical system is not really it's symptom focused, not disease focused in the same way, and so it's actually hard to just count up how many people have Pompe disease that I'm showing you here from EHR records. So to be able to estimate genetic prevalence accurately, we need to know two things. We need to know about the variants that cause disease, and we need to have good reference population databases. And so to look a little bit at the variants that cause disease, and Heidi Ream will likely tell you a lot more about this tomorrow, I'm starting just with looking at the chance of a variant that causes disease. So we call pathogenic variants greater than 99% chance that a variant is going to cause disease, likely pathogenic at 90% to 99%, and then likely benign and benign less than 10%. So there's this huge area of probability in the middle, this 10 to 90%, we're not sure. These are called variants of uncertain significance. And we actually are in the future going to divide these up a little bit. That might be covered tomorrow. And then when we look in clinical databases, so luckily, they're not luckily, like with a lot of convincing and a little arm twisting even, Diagnostic companies have realized that they're able to provide better testing if we all share our data about which variants are pathogenic when identified on the clinical testing that is done by doctors for patients, on patients. And so what we find here is that about 15% of variants are pathogenic or likely pathogenic in this ClinVar database. There's several million variants in here. About almost half of them are benign or likely benign. Overall, if you took like any variant, the vast majority are benign, but not everything gets entered in this database. And actually about 40% are variants of uncertain significance or conflicting, meaning people can't agree on whether they alter the function of the gene or not, or cause disease or not. And all these VUSs lead to about 30% of genetic test reports being inconclusive, meaning the variant's reported and we just don't know if it causes disease or not. When we look at these, when we look at pathogenic VUS and benign and likely benign, and we ask what type of variants are in each category, and this is like not the best color scheme, sorry, but basically all the dark colors, the darkest purple are the loss of function variants. This part in the top is the missense variant, that lighter purple. Basically the VUS that's like 90% there, that's all missense variants. Same for conflicting, that really large portion there. And benign variants, you're getting a lot of like synonymous variants and non-coding variants, and we're like a little less able to comment on the other variants. So that's what we know about pathogenic variation and benign variation. And so how do we look at this in population data? So in NOMAD v4 is a large reference population data set that was released late last year. It has over 800,000 individuals in this data set. It's mostly exome data, includes UK Biobank, and it includes close to 1 billion. I really hoped we had gotten there, but 900 million variants. And it's all built on GRCH38. And it's a vast improvement over the prior version. It's about 5.7 times more samples. And recently, we've been looking at pathogenic variants in this data set. So this is work led by Sana Gudmundsson with some support from Sarah Stenton. And so in looking at, I was just curious, like, do we see more pathogenic variants? Do you see more ClinVar variants in this new data set? The answer is yes. But the answer is also, well, the new data set is 5 times bigger. I hope we would capture more variation in that data set. So we are seeing more variation, and that's on your left is the NOMAD v4, and on your right is NOMAD v2, the older version, comparing the same thing, and everything's a little bit higher in the purple, which is NOMAD, and in the gray is all of ClinVar. All of ClinVar should be the same between the two. And then one of the other things is when we look at the same variants that are seen in both data sets, how do the allele frequencies vary? And it's fairly uniform, but it's pushed to the left. So variants in V4 are a little rarer. These are just pathogenic variants I'm showing you, but if you look at all ClinVar variants, the distribution's similar. They're a little bit rarer, and that is largely because, I guess 2 reasons. One, like, I think this is still a pretty good reference population database, but 2, if you see a variant one time out of like 100,000 people, you don't know if the allele frequency is 1 out of 200,000 for the chromosomes, or if it's actually like 1 out of 1 million or 1 out of 10 million, because you just happen to sample it once. And so it turns out for most of these variants that are rare, they actually are much, much rarer than what we estimated when we only had 100,000 people in a data set. So then this is just showing you the ClinVar variants as a proportion of, like, what proportion of them are in NOMAD in purple versus not in NOMAD in white. And so you can see overall, like, VUSs and benign variants are much more likely to be in NOMAD than pathogenic variants. And this is just looking at pathogenic variants, asking, well, we see pathogenic variants in NOMAD. Are they common? Are they rare? And you can see the vast majority of them are seen only once, not the vast majority, but many of them are seen only once or less than 5 or 10 times. There are some that are actually like really up there in terms of allele counts. Some of that is always like, there are things in databases that are like ABO blood type is marked as a variant that's like pathogenic in ClinVar, but it just means it like alters the function. Obviously having an A versus an O blood type is not a disease. So there are sometimes things like that. There's risk alleles. And then there are things that are just wrong probably in there too. Because these are databases that communities contribute to, and like there's no real screening to make sure that everyone is correct in what they're putting in. And when we look at all the pathogenic variants in ClinVar, you can see it's, the yellow is the autosomal recessive, and that you're much more likely to have autosomal recessive pathogenic variants in NOMAD than all of ClinVar, and there's fewer dominant variants in red. So that also kind of makes sense with what you'd expect in the general population. But why are there pathogenic variants in NOMAD? It's a little bit odd that we're seeing them for even severe dominant conditions. So One is there can be people with rare disease in NOMAD. We do not include any cohorts that are recruited for studying rare disease or the parents from those studies because we'd inflate carrier frequencies. But that doesn't mean, we do include some biobanks, UK Biobank, other biobanks, and that doesn't mean that somebody with a rare disease couldn't sign up for one of those studies, and then get included in NOMAD. However, we wanted to take a deeper look of what's going on. The other situation is maybe there's incomplete penetrance. We're looking at conditions that typically have, they're typically caused by de novo variants, they have very high penetrance, but in some cases, maybe there's incomplete penetrance, and maybe that'd be an interesting thing to study, how some people don't get these diseases. So this is just giving you an illustrative example of one of the cases we found. So this is a condition, so initially we thought maybe there was incomplete penetrance for this variant, this missense variant, GLA45 GLU in, where do I have the gene? In JGB2 there. And so it causes this specific variant. Well, the gene is generally associated with hearing loss. There's a few variants that are associated with this autosomal dominant lethal keratosis ichthyosis deafness syndrome. And so these children normally pass away very early in life, and yet we're seeing individuals in NOMAD with this variant, particularly, there's 34 who are East Asian ancestry and then one in admixed American. And so when we actually looked at the read data, what we saw is that upstream of this really lethal, impaired function missense variant, there is a loss of function variant that is blocking the downstream impact of this missense variant from causing its gain of function effect. And so when we looked at the loss of function variant, that's what I'm showing you here, you'll notice there are 34 individuals from East Asia who also have this loss of function variant. And in V2, not in V4 that I'm showing you here, you can actually look up co-occurrence, which variants co-occur in the same people. It's not as prevalent in V2, but it is everyone who has the missense variant also has the loss of function variant. And that will be true in V4 also. We just don't have that functionality that everyone can see that yet. And so these are kind of situations where there's sort of incomplete penetrance, but we can understand how this rescue is happening. So then we wanted to take a deeper dive into situations like this. So we looked at haploinsufficient genes, that's what HI means, 77 genes where if you lose function of one copy of the gene, it's not enough for wild type function, and these cause very severe autosomal dominant, typically de novo disorders. When we looked at that in NOMAD V4, there were 4,400 predicted loss of function variants. We then used LOFT to filter just the high confidence ones, left us with 3,900. And then we decided, well, we really wanted to look for what else was going on in the genome, so we're just going to look at data or individuals where we already had genome data, which took us down to 734 variants. And when I use the term loss of function, obviously a missense variant can cause loss of function, but I sort of mean it in the classic variant effect predictor sense of nonsense, frame shift, and essential splice sites. So that's what we're talking about here. Just to illustrate an example of some of the things we see when we look at loss of function variation is that this is a variant that we saw. This is from a different study, so this is more an illustrative point, but we have homozygous loss of function variant in this gene that is associated with autosomal recessive Schwartz-Jampel syndrome, where individuals have short stature and muscle abnormalities. It's not impossible, again, that a person could be in NOMAD, but we weren't expecting to see people with this condition in NOMAD. And when we looked in NOMAD, we did see an individual who has a one base pair insertion at a splice donor site that's called as loss of function. And one of the things we look at is other NOMAD or ClinVar variants. So I'm showing you the ClinVar variants on this track that's red there. That's showing you, oh, it is working, okay. So it's showing you that there are loss of function variants, these little X's upstream and downstream of the other variant. So you would expect that to impact the function of the gene. You look at the read data, make sure it looks real. It looks like an insertion there. I think that's pretty convincing. We looked at the NOMAD data. So there was one person who was South Asian who was homozygous for this variant. They were between the ages of 55 and 60 when they enrolled in whatever study they enrolled in. Again, we use LOFT as a loss of function predictor to say if the variant is high confidence loss of function or if there's some concern about it. It passed LOFT, no problem. We looked at the IGV to see what was going on. And so the variant is marked at this site, but what is actually happening is it's inserting an A there that will continue to have the same sequence that will still have the AG acceptor site that you would expect. So obviously, now that we got here and we look at this, we're all like, well, yeah, I wouldn't expect that to be loss of function. But variant effect predictor and other annotation tools didn't realize that. And so some of these loss of function variants that we see in NOMAD are just as simple as that. And so by just looking at the data, just like I walked you through there, we can actually find lots of reasons why variants that are called loss of function in any gene, but particularly in these dominant severe conditions, why they are not actually loss of function. And so we developed a method to do this. And so to walk you through that, there's a whole flow chart that you can go through to evaluate your variants. This is all published in American Journal now. And so basically looks at three things, or has you look at three things. Is there any rescue by a secondary sequence properties? So is there if you have a splice variant, is there a splice rescue nearby? The one I showed you is kind of an example of that. Basically, the insertion rescues the splicing. Is the variant just causing an in-frame change versus like a frame shift? In addition, there can be variants with uncertain biological relevance. One of the things we often found is you would see loss of function variants. This is a dominant condition. You see all these loss of function variants in NOMAD here. They were confusing why they were there, but when we looked at PEXT, which is looks at expression of the gene, it's actually this exon here in black is not actually expressed. It's expressed as these two little exons. And so there are transcripts that have the two little exons, and the canonical has the big exon, but actually the big exon's not really expressed anywhere. And so you can't have a nonsense variant in the middle of a big exon if it's being viewed as non-coding. And the third was just technical artifacts, right? You really need to look at the read data. You can't just go by the call. And so we were finding homopolymers very frequently. There are issues, the other mapping issues that were very clear once one looked at the read data. So we made up a bunch of rules. There's a whole thing, 32 rules you can go through. But the bottom line is when we looked at this for those genes that I told you about, those 77 haploinsufficient genes, we found that 70% of the variants, just by looking at them, we could say did not result in loss of function. There were some in gray where we're actually not really 100% sure. Sometimes that was because we didn't actually have the read data. We don't have it for everything in NOMAD, and we didn't want to make a conclusion based on incomplete information. But there were some that we thought still looked like they would cause loss of function. In a different study, we had looked at a number of recessive conditions, actually that prevalent study I started talking about. And so there we saw actually 27% of variants still, and even in recessive conditions, were not loss of function. And there was a whole number of reasons why they were not loss of function in this dominant condition. Again, PEX was very common, again, it being a loss function variant in a part of the gene that wasn't really expressed. And then last exon, we just treated a little bit cautiously. It's hard to know how to interpret those without more functional evidence. So basically, we had 170 predicted loss of function variants left at the end of this that we were interested in pursuing. And one of the first things that happened is we started looking at what can we do? Can we know anything about these cohorts? And NOMAD includes 1,000 genomes cohort, which is an open access cohort. And so we know you can look up those individuals because the data is available. And so we're like, wow, there's a lot of variants in this 1,000 genomes cohort. We know a lot about these people. This is going to be great. And then we were like, oh, these are cell culture artifacts because these are really well studied. And so these get grown up in cell culture over time. And so we're just acquiring somatic loss of function variants as culture artifacts. So we had to throw those out. Then we looked at what was left, and we actually were realizing there was a lot of clonal hematopoiesis-associated genes here. So we looked at variants there. We were a little more stringent in our allele balance cutoff there, and we got rid of a bunch more. So we're left with 50 loss of function variants. We think it'd be very interesting to study these individuals. NOMAD isn't the best place to do this because it's not consented for individual study. It's a summary cystic database. But doing these kind of things in all of us or other biobanks, we think could be very interesting. I'll leave you with that. In my last few minutes, I will get to the actual title of my talk, which is about the GREGoR consortium. And so I just very briefly wanted to tell you about this consortium that involves 5 research centers and 1 data coordination center that's all funded by the NHGRI that is working towards improving technologies to understand rare disease diagnosis. So we've been recruiting samples from many different countries, including in the US, and we've had a lot of efforts to really address disparities through this, such as having consents in foreign languages, having remote collection of samples, so people who are not necessarily at a major academic medical center are able to participate in genomic research. We're trying to work with clinics for uninsured or underinsured and doing things to try to reduce the VUS disparity. We work with collaborators from all over. And so some of our goals are to create a data set of broad utility that we're really hopeful the community will use, which is part of why I wanted to tell you about it today, to increase the diagnostic yield, but not sort of case by case the way UDN does, but really by applying a number of different technologies and figuring out which technologies and which approaches are going to help us understand functional variation that causes rare and human diseases. And then from this, try to develop genomic recommendations for the larger community. So we have individual level genome, phenotype, and pedigree data, all shared through ANVIL, which is a controlled access data set. So it's on the cloud where you actually bring your tools to the data as opposed to trying to bring all the data locally, because these data sets are getting really big, and so we don't want to store endless copies of them. It's a mix of consent for general research use and health and biomedical use, but that makes it very broadly consented for research. And we're working, and I'm going to show you a hint of some open access genomic summary results that we want to make this data set very accessible to everyone. We put all our candidates and our variants, for new genes anyway, into Matchmaker Exchange, and everything we found is also included, in terms of like genetic diagnosis, is included in the ANVIL data set. And we're depositing variants to ClinVar and sharing new genes and the relationships of genes with disease with the GenCC Gene Correlation Coalition, which Heidi will also tell you more about. So this is not available yet, but many are actively working on this, and the idea is to just be able to go in and type in any gene or a variant, and be able, or coordinate region, and look at what variants we're seeing in this gene, and we're going to add some more annotations. What variants we're seeing within the GREGoR data set. So if you had a gene of interest, you could see what variants are in it, and then you would know if you wanted to apply for access to learn more about this data. So that will be something that will be more open access. We also have put the GREGoR data set onto Seeker, which is a genomic analysis platform that is available to anyone to use on ANVIL that has exome and genome data that they want to do rare disease analysis, like family by family rare disease analysis. We have many people from external to the GREGoR consortium come and use this, and many of the groups in GREGoR are also using it. Helps you project manage and do the analysis. And it also is a data sharing platform, goes right into Matchmaker Exchange. And so currently you can look up on Seeker. You can look up on Seeker any variant. So I'm looking up one of those variants from that prior list of the gene I had shown you on the other page. And you can see here it's a 5' UTR variant. You can see some in silico scores. You can see how common it is in Nomad. You can link out to all these things. And then the initial access will be, for people who do not have data in Seeker, the initial access will be just that you'll be able to see the genotypes and whether they're affected or not. So that will be somewhat helpful. For those who have full Seeker access, you can see all the HPO terms that the individuals also have. And we're working on like an intermediate level where you can maybe see like the organ systems, but not the like full HPO descriptions for those who don't have full access. But so it'll be a way to kind of go in different ways and be able to see the data and again decide if it's worth your time to access it even deeper. We worked on a really complex data model to collect all the family, participant, and analyte information. We're really hoping that others adopt some of these standards so that more data is interoperable. And so this is all available, again, all our data is in this format, and then these formats are available on the GREGoR website, which is just gregorconsortium.org, I believe. And then this is our data set thus far. We're about 3 years in, and it's a growing data set. We contribute data about every 6 months. And so it's 4,000 participants from 1,800 families. There's a lot of short read data and short read RNAs growing, and then there's long read data from 157 participants, and there'll be more and more over time. There's also there's some ATAC-seq that's going to go in there. We're trying some proteomics. There's a whole bunch of different omics that are going to be in there too, some metabolomics, I believe. And so it should be a really fun data set to play with. And these are all rare disease patients that are waiting for diagnoses. So anything that we do find, we're able to give a diagnosis back going through one of the GREGoR centers. We have a lot of cross-consortia collaborations from GREGoR across to other consortia. So we work with IGVF a lot, which probably many of you have heard of and know more about than I do, but just that we're collaborating in a number of different ways. And Moaz Dawood, who is here, has this recent preprint that really shows that when you have MAVE data on different data types, and thinking about VUSs, it really just reduces that disparity. There's a lot of reasons for the disparity we're seeing in VUSs, but this is obviously a big problem because it means that people of different ancestries, when they get genetic testing, are kind of getting a different chance of getting a diagnosis. We've done a lot of different analyses, which I'm going to go through very quickly. So we looked at what do you find when you do genome sequencing after exome sequencing. We solved a lot of cases that could have absolutely been diagnosed with exome reanalysis, but it turned out we were actually missing a lot of things in structural variants, and then there were a lot of regions of coding variants that were poorly covered in the original exome sequencing that we needed the genome sequencing to diagnose. And so both in this cohort and a replication cohort, we were able to uplift the diagnostic rate with short-read genomes by 8%. We looked at copy number variants. We had another 2.6% uplift. Mitochondrial variations was 0.4. SMA, spinal muscular atrophy, we had 11 diagnoses. We've been doing expansion hunter, a number of other diagnoses. We looked at somatic variation. We found just 2 candidates in that, and then proteomics and long read are coming. I'm just going to take 1 minute, sorry, to explain just really two really cool non-coding diagnostics. I'll go through this really quick. So the first is a deletion that was found in the gene chaser. It took us 5 years to find 3 patients with this condition because it's a non-coding RNA that is, you need a SV deletion for, so you need to both have genome data and you have to know to look at a long non-coding RNA. And this is work led by Vijay Ganesh in collaboration with Gemma Carville. And these are the patients there and their brain findings. But the important thing is chaser, what it's doing is regulating CHD2 negatively. And when you delete chaser, you're upregulating CHD2. So in these patients, we can see upregulation of the protein in the Western blots. And this is happening on the allele that is in cis to the deletion. So this was one of the few non-encoding RNA that's now been linked with rare disease. This was the craziest story I've ever been involved with. This happened over like 5 weeks. Yu-Yang Chen and Nicola Whiffen from Oxford reached out about a variant that they were seeing that was in a depleted region of NOMAD, but they thought maybe it was an artifact, but they were seeing a lot of patients in the Genomics England data set who had a variant there. And so we have 119 patients collected over 5 weeks that all have basically this insertion of a base right here. Sorry, this is like a little hard to do, that is impacting like this turn here. And this is one of the most common causes of syndromic intellectual disability that's been completely unrecognized until now, another non-coding RNA diagnosis. So I'll just make a last pitch that missense variants are really hard for us to interpret. We have lots of VUSs and missense variants, both within our GREGoR cohort and the larger cohorts. And so we're really interested in collaboration or other ideas you have to be able to improve diagnosis. And just thank the whole team and all of the whole GREGoR consortium, which I forgot to put in the acknowledgement slides. But thanks everyone for your time today.
Okay, thanks very, very much. That was really super interesting and deep, and we're thinking about, I've got loads of questions, but I'm going to hold mine back. So, questions from the audience? Got a few minutes. And don't forget, I think there's somebody up there, don't forget to say your name. Maybe that's useful.
This is Moez, whose data I showed a little of.
Moez Daly from the College of Medicine. So I kind of asked this question to a lot of people. Of all the molecular diagnoses that are going to be out there for individuals with rare disease, what percent do you think are... Is it on now? Awesome. Sorry about that. Of all the molecular diagnoses that are going to be out there for individuals with rare disease, what percent do you think are going to be coming from coding variants versus kind of the high penetrant, large effect, non-coding variants? And then as the follow-up, do you think essentially that there is a treasure trove of high penetrant, non-coding variants in these genomes that we have unsolved?
I personally don't think there's a ton of non-coding variation in terms of finding diagnosis for rare disease. I think a lot of the non-coding variation has slighter effects and perturbations, and so isn't going to often be as strong. When we look in the NOMAD data, getting back to it, we see genes that are constrained for loss of function. And of those, there's about 3,000, and of those, still about 1,200 or 1,500 of them do not have rare disease, any disease association yet. And so maybe in those where there are very dosage sensitive, those are places we should be looking in non-coding. CHD2 is one of those genes. Obviously, CHD2 is already associated with a disease, a different disease and different phenotype than the chaser deletion. But I don't, for some reason, I have the number of like 5 to 10% in my head of the undiagnosed patients would be non-coding at the most. I think a lot of this is still coding variation around known genes that we don't recognize, or the function of it, and then I think there's a lot of gene discovery still to be found.
Thank you.
Hi, Bill Mowry, Alexion Pharmaceuticals. It was a really fantastic talk. I was particularly interested in some of the instances that you cited of interactions of variants that are in cis to each other, or how they change the interpretation of potential loss of function and that sort of thing. How prevalent do you think these effects are, and do you think it would make sense to move to a model where we aren't considering just single variants in isolation and thinking about like the haplotype of the gene, or the entire set of variants within one copy of the gene?
I think there is value in doing that. I think it increases the number of things you need to look at almost exponentially very quickly. And so I don't think it's wrong to take a single variant approach to start, but I think we are going to need to study variant combinations. I think in IGVF, that's one of the things they are focusing on, is making sure some of their effort goes to combinations. But they're probably, I can't speak for them, but they're probably still in the 2 or 3 variant spaces, not in like large haplotypes. But there's still obviously a lot to find, and so starting with more simple things, and then we have a ways to go. And I do think we're gonna have to study combinations, and that's some of what we're missing.
Okay, so JT, I'm going to abuse my position now, since you did earlier. So I was thinking about what you said about the 5%-ish intuitive guess, and this question which was connected to it. And I was thinking about ascertainment bias in the sense of, when we look at these summary statistics, we're looking often at things that are summaries and singles, and we aren't looking in whole regions outside of the huge structural variation. And so there is still a lot of unexplained, as I understand it, rare disease, and it's not clear to me that it would necessarily be as low as that if
I hope
we
it's
had
not.
models that could find those
Yeah.
larger changes that we know are more common in those regions.
Yeah, I would love non-coding variation to explain a larger portion. I just haven't seen a lot of evidence for it. And it could just be that it's very hard to interpret and that we just need more time and more approaches. But I don't know. It's just,
Maybe that motivates a consortium having permission to do more than the summary statistics.
I mean, I think IGVF is actually very focused on non-coding, so hopefully things will be coming out of that.
JT, did you want to?
Yes.
Or am I?
So where do you think, in the short to medium term, in vitro functional data is likely to have the greatest impact, in a sort of prospective or retrospective mode? So should we be going out and making lookup tables of everything, or are these things going to be so tissue and condition and stimulus specific that it's better to have model systems that can be quickly deployed and genome editing tools that can be quickly deployed?
I mean, first off, I would love if the data that exists is easier to look up. And I know that there are a lot of efforts in that space, and a lot of people in this room involved in those efforts. But we come across variants all the time as we're analyzing cases that we can't spend a day or two digging into a data set we don't even know has the variant. And so having ways we can just look up and figure out if our variants are there is just a lift that then, for the existing data, makes it more utilized. But the vast majority of variants that we would encounter that we're interested in, we will not be in these databases. So I would say the second is going to have a larger impact for us. I just don't know that that's necessarily how you want to approach it. And it is, I think, easier for me to interpret a variant if it is in a large experiment than if you just CRISPR'd a few variants in a gene where I had like one that I was interested in. Those are hard to interpret, right? Like what do I make of that? Whereas when you have these deep mutational scanning, that's fantastic because I can see the variants and where my variant falls in terms of what's going on with the rest of variation in the gene.
Okay, well, thanks. And again, that was wonderful. And